Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello, welcome to class. Let us discuss now the process of recombinant DNA technology, the last two steps. That is, the sixth step in the recombinant DNA technology is growing of a recombinant cell on a large scale to get a desired product. That is, growing of recombinant cell or a recombinant host, we can say. That means it has the host which has the heterologous gene will be called the recombinant host, and this host should be grown on a large scale to get the product of our interest. Okay, on large scale. And this large scale production is possible only when it is grown in, when the host is grown in large vessels. And these large vessels which are used to grow the host cells on a large scale are called bioreactors. The bioreactors are majorly applied for growing the host cells on a large scale, especially for animal and plant cells. Suppose if those large containers are used for growing the microorganisms like bacteria, then they are called fermenters. So that is the only difference. Here the controlling systems, everything will be same, but if the vessel is used for growing the plant or animal cells, then they are called bioreactors, and if it is used for growing the microorganisms, then it is called fermenters. Then this development of fermenters or the bioreactors is based on the nature of growth of the organisms. For example, all the living organisms, especially the microorganisms, when they grow, during their growth, they exhibit different phases in their growth. For example, if we plot a graph like this, then by taking a nutrients available on one axis and growth on other axis, then initially the organism will be, when it enters into a bare place where the nutrition is available, initially it will start adjust for the environment. Okay, and that phase is called lag phase. That means it is not showing much growth, but it is getting adjusted for the environment. And after getting adjusted for the environment, it is ready to take. It will synthesize all the enzymes and growth factors for its growth, so that it can utilize the nutrients on a very fast way, and it will grow very fast or divide very fast, and that phase is called log phase or exponential phase. And this will happen till there is the availability of nutrients. Till the nutrients are available, then till that time the organism will grow on a fast rate and produces the product from this. That is, that organism, whatever the product possible by it, it will produces and utilizes the nutrients. Once the nutrients are getting exhausted, and the cells will start digesting the other cells, and of course the cells will start producing some toxic substances. Then at that time the environment will not be suitable for the growth or division of the organism. At that time, for some time it will be on a stationary phase. That means this is called stationary. The organism will not be growing if the nutrients are exhausted. And after this phase, the toxicity will get still more increased because of the metabolic waste and all, and the cells will start dying. The cells will start dying, and that is called death phase. Biotechnologists will look for at which phase we could obtain the product on a large scale. It is at the lag phase or the exponential phase. So by studying this, there is a development of two types of bioreactors. There are two types of bioreactors where we can grow the organisms. That is microorganisms, it may be plant or animal cells on a large scale. So this is based on the phases of growth available in the organisms. And those two are, the first one is fed batch bioreactors or fermenters. Fed batch bioreactors, or we can call fed batch bioreactors. And the second one is continuous bioreactors or fermenters. Let us discuss one by one. Fed batch. The first one type of bioreactors are fed batch bioreactors, and in that there are two types. One is stirred tank bioreactors, and the second one sparged bioreactors. Bioreactors, they are available in various sizes, that is ranging from 100 to 10,000 liters capacity. They are available, and usually they are made up of stainless steel because it is user friendly. It will not be rusted very fast. So for that reason, the bioreactors usually they are made up of stainless steel. Of course, there are other materials too used, that like porphyrin, but preferably stainless steel, and they are available in the sizes ranging from 100 to 10,000 liters. And any of the bioreactors will have the systems of control. For example, there are controlling systems for controlling a pH, controlling temperature, controlling nutrients, and controlling oxygen. If it is a aerobic type of organisms, then oxygen control will be there. If there is an anaerobic organism, of course there will be oxygen control where the oxygen should not be supplied. So all the controlling systems will be there for the bioreactors. And let us look at the stirred tank and the sparged bioreactors, a type of fed batch. So here in stirred tank bioreactors, it is a tank where it is enclosed, and there will be a shaft given. The shaft will be connected with a motor and a gauge, measuring gauge for that. And this shaft will have the blades. The blunt blades will be there. So like this, many blades are provided to this. So this will rotate like this, and there will be an opening, and there will be opening for pH and pH control, and there will be entry for water, so that it control the temperature. During fermentation process, there will be increase in the temperature. So to regulate that, the cold water will be supplied surrounding this, so that the temperature will be cooled down. So here in the still tank bioreactor, we are calling it as a fed batch because the bioreactor will be cleaned once, and into this the nutrients, usually the nutrients are added in the form of broth. Broth means the liquid. The nutrients are given in the form of liquid in nature. If it is given in the solid form, then it is called agar, medium. So, but in fermenters or bioreactors, usually they make use of the nutrient broth. So, nutrition will be given in the form of broth, that is in the liquid form. And into this, first the nutrients will be filled in the form of broth, and to this the recombinant cells are added, and the shaft or the stirrer will be there. It will be go on mixing it. So when there is a mixing, the nutrient will have the proteins on large amount. So upon the digestion of proteins, there is a creation of foam. So this foam may create some problems for the fermentation process. So there will be a foam controlling system. So there is a foam breakers. So foam breakers will look after that. Okay. So here, once the nutrients are added, the organism will be added, or the recombinant cell will be added, and it will be allowed to grow for defined time period. For example, 1 day, or for 6 days, 7 days, depending on the type of product. Okay, so here the nutrients will not be taken out in the middle. Okay, if it is set for 1 day, after completion of 1 day only, the entire nutrient will be taken out at once, and the product is purified. So that's why it is called fed batch. The organisms are grown in a batch. They are not grown continuously. Okay. So here there is a stirring system for mixing of all the nutrients equally. We are calling it as stirred tank bioreactors. So same way sparged bioreactors are there, where sparged bioreactors will have the blades and the sparger. There is a presence of sparger. These spargers will give the oxygen in the form of air bubbles. So these air bubbles, when they created, they will increase the surface area. So they increases the surface area. Everything will be same. Controlling systems, everything will be same. Only the difference is for mixing of the nutrients and equal supply of oxygen, here there is a stirrer available. But here there is a sparger available. So where the sparger will give the oxygen in the form of air bubbles, and of course they increases the surface area so that the organisms which are introduced into it will utilize the nutrients on a large scale. So that is, they will be grown in a batch, in one batch defined. For example, 100 liter of nutrient is added, 1 liter of growth organism will be added, and it will be grown for 1 day. After 1 day it will be harvested. So that is called in a batch they will grow. There are systems available where in a continuous type of culture, the organisms are maintained at one phase. Look here, in fed batch, the organism will complete all of these phases. First, initially it will be in lag phase, and it will enter to log phase where it will produces large amount of products, and stationary dead phase. All phases will be there in fed batch. Whereas in continuous, the things are different. Okay, so we shall explain now what will happen in the continuous bioreactors. So here I'll write here. In continuous, the second type. In continuous type of bioreactors, it is an open system. So consider it as a bioreactor. So there will be one opening above and there will be opening below also. So here it is for addition of nutrients. It is for addition of nutrients, and remember nutrients will be given in the form of broth here also. It is outlet for taking the products. And of course here also temperature, pH, foam breaker, everything will be there. Controlling systems, all of will be there. I am not going to explain this again here. But the thing is, in this system, in the continuous bioreactors, the organisms are maintained at the organisms. So whatever the organism, recombinant cell we add to this will be maintained at metabolically active phase. The metabolically active phase in the growth of organism is log phase or the exponential phase. So they will be maintained at log phase. You may ask the question, how it is possible to maintain them at a log phase? It is possible only when the nutrients are added continuously. The organism is not allowed to enter into stationary phase. Here we have plotted graph here, right? When the nutrients are getting exhausted, then only the organism will enter into the next phases. Otherwise, no. Okay, so the organism is allowed to reach the log phase first. Once it reaches the log phase, the nutrients are added continuously on a defined amount. So a large amount of nutrients should not be added. In a defined amount, the nutrients are added to the reactor continuously, so that the organism will go on producing the metabolic products, and it will not enter into the further phase. So whatever the products produced will be taken out. But the disadvantage of this is, since it is an open system, the nutrients are continuously added, there are more chances of contamination. While adding the nutrients, there may be other organisms enter too. For that, the quality checking will be done for defined at every intervals. But it is not required in case of fed batch. Whether we have to choose fed batch or the continuous will depend on type of products. For example, if they are making the products like wine, they make use of fed batch bioreactors, not the continuous. Okay, so this is about the bioreactors, the two type, fed batch bioreactors and the continuous bioreactors. And the next step after growing the organism, the recombinant cell on a large scale, the next step is the isolation and purification of the products, and that step we call it as the seventh step in the recombinant, and it could be the last step, is called, together we can call it as downstream processing. Downstream processing. Till here it is called upstream processing, starting from the isolation of gene and cutting off that desired gene using restriction enzyme, cloning it into a vector, inserting into a host and growing the host on a large scale in bioreactors. Till then it is called upstream, that is the product is produced, and after production of the product completely, that product should be purified from other impurities. For example, if we grow some product here, say for example wine or any insulin for that matter, not only wine, not only insulin is present. Along with that, it has the growth organism, that is the host cell, some toxic substances produced from the host, some metabolic waste will be there, nutrient waste will be there. So everything should be separated and the product need to be taken out, and those the process used for used for process or the techniques, processes and techniques used for isolation and purification of product is called downstream processing. For example, using gel electrophoresis or by using chromatographic techniques or centrifugation techniques, any of the techniques, the product has to be separated or it should be isolated. After isolation and purification, next it will be sent for quality checking and clinical trials. Clinical trials are required in case of drugs, that is medicines, for example, in case of insulin or any other drugs for that matter, there is a clinical trials. The clinical trials, so first on a first step they will be done on animals and later the trials will be done on human volunteers. After that only the product is allowed for marketing. So these are the steps used or steps to be followed in recombinant DNA technology. And there are some additional things we should know about biotechnology, like DNA libraries, molecular probes, blotting techniques. So let us discuss them in the next class. Thank you.